Hello students, welcome back to Pharmaceutical Concepts. Let us learn one important topic from biochemistry today, that is enzymes. Why we have to learn enzymes? What is there in enzymes to learn exactly? Enzymes have various therapeutic applications. For example, some enzymes they possess anti-inflammatory property. There are some enzymes possessing even they are used in cancer therapy also. Okay, they are used in cancer therapy also. Next application, enzymes also have certain analytical applications, means some enzymes are used as analytical reagents for estimation of compounds. For example, cholesterol oxidase. Okay, whenever there is ase means that is the enzyme. Okay, so cholesterol oxidase is used for estimation of cholesterol. There are applications of enzymes in genetic engineering also. For example, restriction endonucleases are the enzymes which are used in gene transfer and DNA fingerprinting. There are DNA polymerase enzymes which are used in PCR, that is polymerase chain reaction. Synthesis of variety of products used in food industry. For example, the alpha amylases. Alpha amylases, they are used to convert starch to glucose. Even enzymes, they have diagnostic applications. These are involved in many body processes and regulations. Digestive system, for example, includes many enzymes like lipases, amylases, proteases. And the diagnostic applications which I was talking, that is for detection of many diseases, means for detection of many diseases. For example, alkaline phosphatase, they are used for the detection of rickets. Similarly, the gamma glutamyl transcriptase, similarly aldolases, they are used for the detection of muscular dystrophy. Okay, so there are wide range of applications of the enzymes, and thus we have to learn the details of enzyme. We have to learn the structure of enzyme, what is and where is the active site on enzyme, how these enzymes work. By definition, enzymes are biocatalysts. They are synthesized by living cells. They are proteins in nature. We know all enzymes are proteins in nature, but all proteins are not enzymes. Okay, so what is the difference between protein and enzyme, that we will learn here. Enzymes have certain characteristics which are not there in proteins. Okay, so all enzymes are proteins, but all proteins are not enzymes. Enzymes are also colloidal and thermolabile in character, and they are specific in their action. Okay, enzymes are specific in their action because of the active site present on enzymes. So enzymes are biological catalyst. Catalyst means what? Catalyst is any substance that increases the velocity or rate of chemical reaction without itself undergoing any change in the overall process. So just the presence of that particular catalyst speeds up the reaction. And since enzymes, they speed up the biochemical reactions, various biochemical reactions like digestion, absorption, metabolism, and there are biological oxidations and so many reactions which are happening within the body. For them, enzymes help in speeding up. Enzymes are 3D globular proteins. Yes, enzymes are proteins in nature, but they are 3D globular proteins. If you remember the conformation of proteins, primary protein structure, secondary protein structure, tertiary and quaternary protein structure. Okay, so proteins are what? Proteins are nothing but they are made up of amino acids. So amino acids, they come together, they make proteins, and such many polypeptide chains they make enzymes. Okay, so it's a long chain of amino acids. For example, all these amino acids, they are chained in one line. They have made protein, and this protein, when this chain is just in the form of, you can see here some loops are there, some coils are there. So these all chains of amino acids, they have coiled up, they have made certain loops, alpha helix, beta sheets, and this 3D form of the protein is called as enzyme. So enzymes are 3D globular proteins. You can see here in this particular video, the 3D form of the enzyme is clearly visible. This enzyme is nothing but enzyme lipase, and I have taken this from the PDB, that is the protein data bank. So this is the nothing but the enzyme. Each enzyme has its own tertiary structure. Each enzyme has its own tertiary structure and specific conformation, which is very essential for its catalytic activity. So this, what does this mean? That specific conformation and own tertiary structure means the sequence of amino acids which are present in the particular protein or in the particular enzyme that defines the protein, the enzyme, and the specific action which is represented by that particular enzyme. There is a specific region on the enzyme which is called as active site, where substrates, cofactors, prosthetic groups, they bind. So on each enzyme there is something called as active site. This is the 3D protein or the 3D enzyme structure which we have seen just now. So for better understanding, we usually draw enzymes in this way. So this is the same enzyme I am drawing in 2D format, and you can see here this is the substrate which has to bind on this enzyme so that some biochemical reactions will start. So where exactly this substrate is going to bind to this enzyme, that is at the active site. So this is the active site of the enzyme where this substrate will bind and the reaction will start. So you can see here the active site has two regions, that is binding site which selects the substrate and binds it. So it is very specific to the substrate, just like lock and key. Any substrate, any structure cannot come here and bind to any enzyme of the choice. No, it is not like that. The site means what? This is nothing but the amino acid. No, we have seen many amino acids, they were in the form of chain, and this chain was then just made in the form of loops. So the amino acids which were far here, maybe they are just adjacent to each other in the form of the loops and helices, right? So that makes the active site. So if I call this particular region as active site, so this particular region is made up of the amino acids which are present in the vicinity. The second red color site which you see here, that is the catalytic site, which reduces the chemical activation energy. So binding, this active site occupies less than 5% of the total surface area. It has specific shape and change in shape because it may affect the function of the enzyme. The active site consists of amino acid residues that form temporary bonds with the substrate at the binding site and residues that catalyze a reaction of the substrate at catalytic site. So you can see here the substrate binds here at the active site and the reaction starts. Whole enzyme, that is the whole active enzyme, is called as holoenzyme. This holoenzyme is made up of protein part and non-protein part. So this binding site, you can see here, this particular region where the coenzymes and cofactors, they will be binding. Okay, that is the non-protein part. That is, these are the non-protein parts of the enzyme. So this non-protein part is nothing but coenzyme, and the protein part of the enzyme is called as apoenzyme. And the term prosthetic group also will come somewhere when you will read this chapter. So prosthetic group is used when the non-protein moiety tightly, that means covalently, binds with the apoenzyme. That is the prosthetic group. So here we have seen just now the enzyme reacts with the substrate and it forms enzyme substrate complex. A reaction will begin. Substrate will undergo some changes and it will leave the enzyme as a product. So this is the product formed by leaving the enzyme available for reuse. The product P is released after the catalysis and the enzyme is available for reuse. So this enzyme and this enzyme, the same, okay, it is the same enzyme to which the substrate was attached. What does this binding mean? What does this attachment means? Means the various interactions occurring between the amino acids of the active site and the substrate, the enzyme which we have seen just now and the substrate which is binding in this particular site. When the substrate comes here, there are various interactions happening of the substrate with the various amino acids which are making the active site. What are these interactions? These interactions are in the form of hydrogen bonding, hydrophobic interactions, ionic bonding, and some temporary covalent interactions like van der Waal interactions. These interactions form the enzyme substrate complex. Enzymes don't always work alone. They get help from cofactors and coenzymes. We have seen just now, that is the non-protein part of the enzyme. Non-protein part of the enzyme. So what are these coenzymes and cofactors? Let us see. Coenzymes are low molecular weight organic substances. Non-protein part, no, both of them are non-protein in nature. Coenzymes are low molecular weight organic substances which are loosely bound to the active site of the enzyme. They help in proper functioning of the enzyme. Coenzymes are also called as secondary substrates as they have similar affinity as that of the substrate. There are certain characteristics of coenzymes, that they are loosely bound to the enzyme, they are thermostable, they help in biochemical transformations, and these are some of the examples of the coenzymes, that is nicotinamide nucleotides, biotin, ATP, etc. Let us see what does cofactors do. What are cofactors? These are also non-protein, we have seen. These could be organic or inorganic molecules and they bind either loosely or tightly. So based on their loose binding or tight binding, again there will be subtypes. They can be removed by denaturing the enzyme and its presence is essential for the activity of enzyme. That means they help the enzyme to carry out their function. We can also call them as activators of the enzyme. So certain examples of the cofactors are metal ions like sodium, copper, cobalt, zinc, potassium, and so on. Moving on to classification of enzymes. First,